Good morning, everyone. Thank you for attending the Masonic Cancer Center seminar series. Today's speaker is Dr. Zohar Sachs. She is an assistant professor in the Department of Medicine, Division of Hematology, Oncology, and Transplantation. Dr. Sachs received her undergraduate degree in chemical engineering from MIT, Cambridge. She then attended Tufts Medical School and did her MD and PhD program. Dr. Sachs completed her residency in internal medicine at Caritas St. Elizabeth's Medical Center, Boston. And then she moved to University of Minnesota to do her fellowship in hematology, oncology, and transplantation. She joined the faculty as a physician scientist here at the University of Minnesota in 2012. She is a very active translational scientist and has a active basic and translational research lab. And the lab focuses on molecular mechanisms around the self-renewal of acute myelogenous leukemia. Dr. Sachs specializes in the care of patients with hematological malignancies. And today she is here to present on mechanisms of progression and aggressive biology of TP53 mutant AML. Thank you, Dr. Sachs.
Thank you. Thank you so much for everyone who's actually here in person. It's a very, very cold day, so I appreciate anyone who was able to come. So thank you, and thank you everyone who's here on Zoom. This is my favorite seminar, the Masonic Cancer Center seminar, so it's really an honor to be here. Let's see. Oh, there we go. Okay, so I think, if any of you have heard me speak in the last few years, you would have seen this slide, but I never get tired of this slide. And this really highlights the fact that the work that we do, the work that we do as a cancer center, as lab-based researchers, is actually having a major impact on the care of our patients. So as Deepa mentioned, I'm a clinician as well. I take care of patients with AML. And when I started my faculty practice 10 years ago now, I was using the same chemotherapy for AML that we'd been using for like 3 decades. So we really had nothing new to offer at that time. And then just in the last 5 years, we've had this big list of drugs that you see. Those were all FDA approved in the last 5 years. So we had nothing for decades, and then bang, 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 all these targeted drugs. And these are all drugs that are targeted towards molecules that were identified through basic research, basic science research coming out of labs. So the molecular revolution in biology has yielded us these countless of new cancer therapy targets and cancer drugs, and they're really effective. Patients with AML now have options when they relapse. They live longer, they live healthier lives, they're enjoying their time. And this is true across pretty much every cancer that I treat as a blood cancer specialist. And it is also very true for the solid tumors. So this is a really exciting time to be taking care of cancer patients. Unfortunately, there is one subtype of acute myeloid leukemia, AML, that the molecular revolution has not impacted, and that would be AML with P53 mutations is refractory to all those drugs that I showed you. So here I have a survival curve that shows you that if you have a P53 mutation, look at this 50% survival. So the median survival here is on the order of a few months, and very few patients, look at that, very, very few patients make it past a year, and almost no one makes it past 2 years. So this is a very treatment refractory AML subtype where that's almost invariably fatal. The trick is P53 mutant AML even though, the outcomes are so poor, like I showed you, it actually does respond to therapy up front. So we can, with, traditional chemotherapy, achieve something like a complete remission, or at least really good disease control in P53 mutant AML using our standard drugs. The problem with this disease, and actually really any AML, is that the patients relapse. So despite really good upfront disease control, almost everyone relapses with treatment-resistant disease. And so it doesn't take a scientific genius to come up with a solution. That the problem with AML is relapse. And so why does AML relapse? And relapse is caused by cells that can recapitulate the disease, and those cells are called leukemia stem cells. So leukemia stem cells are the cells that cause relapse and death in AML and many cancers. And the leukemia stem cells are defined by that capacity, the ability to recapitulate disease. These are the cells that persist after therapy. Experimentally in the lab, we define a leukemia stem cell by the ability to recapitulate leukemia in mouse. And we also use cell surface markers to identify leukemia stem cells. So those are in humans, CD34 positivity, CD38 negativity. But this is really kind of an imprecise label. So it sort of helps us find a leukemia stem cell, but it's not a perfect label. And what do we know? Just from what I told you earlier, we know that a P53 mutant leukemia stem cell is uniquely treatment refractory. And despite this being a massive, probably the top clinical problem in the care of AML patients, P53 mutant leukemia stem cells have not been characterized. There are almost no publications on P53 mutant leukemia stem cells in human models and in human disease, and very, very few publications dealing with this in mouse models. Okay, so that whole kind of state of affairs got us interested in P53 mutant AML, and up until I would say up until a few years ago, when I had a young patient with P53 mutant AML who, I wanted to figure out what to do for him, and I looked at all the papers and all these new drugs, and I thought surely something could work, and I came to see that, in fact, nothing would work for this guy. Nothing would be predicted to work, and despite our best efforts, he lived about 5 months from his diagnosis. And so I thought, well, in our lab, we study leukemia anyway. Why don't we think about P53 mutant leukemia stem cells? And it was really this fact, this realizing that P53 mutant leukemia stem cells haven't been well studied. And up until that point, our lab was really focusing on leukemia stem cells in general, that we thought, well, we can study this field. So that's why I want to tell you a little bit about what we did as a lab coming in before we started focusing on P53 mutant AML. So we really focused on studying mechanisms of self-renewal in AML. And what we did previously was really trying to understand the molecular pathways that allow for self-renewal in AML. And just to summarize, I don't even know, countless years worth of work. We basically performed single cell RNA sequencing on the leukemia stem cell compartment, because like I told you before, leukemia stem cells are functionally defined, or they're defined by cell surface markers, but we don't really know which cell is a stem cell and which one isn't. And so I thought single cell RNA sequencing could help us figure that out, to figure sort of the functional state of that leukemia stem cell. And basically what we did was take leukemia stem cells from a mouse model. We did single cell RNA sequencing. We looked at the gene expression data, and we found these 3 distinct gene expression profiles. We looked for cell surface markers of these gene expression profiles and found that CD69 is very differentially expressed between these cell surface proteins, gene expression profiles. And so we sorted based on CD69 and some other markers, and found that it was only the subset of leukemia stem cells that express CD69 at high levels. So here we look at gene expression, but here we're sorting on protein. So high CD69 protein sorry, I can't see well. This is the high CD69 protein. So the leukemia stem cells that have high CD69 protein expression, and we're missing our arrow here, I apologize, but basically those were the cells that were able to recapitulate leukemia, defining self-renewal capacity. And among the stem cells that have low CD69 capacity, they are unable to recapitulate leukemia. So in so doing, we identified a novel marker of leukemia stem cells that's more precise than what the field had. But I felt that, we felt that what we were doing was kind of broader than that, because we were able to define the single cell gene expression profile of self-renewal, and therefore define a functional state of self-renewal at the single cell level with really good precision. And so we felt that CD69 is not just another cell surface marker, but it is reflecting a functional status of the cell. And then we went on to look deeper into the gene expression profiles, and we saw that this gene expression profile is highly enriched in NF-kappa B target genes. We know that NF-kappa B has been implicated in leukemia self-renewal capacity, what was the, and CD69 itself is an NF-kappa B target gene. Yes, Carol.
Before you go too far down the track, how did you purify these cells? Was it by that marker, or was it by the ability to biology?
Excellent point. Yeah, thank you for asking that. So the first thing we did, so previously, before we did this work, we identified the cell surface protein expression of the leukemia stem cell enriched compartment of this mouse model. Okay, so we had like sort of the standard stem cell markers, and we demonstrated that that is the stem cell both in mouse in vivo recapitulating leukemia recapitulation assays, and also in vitro with like serial colony forming assays. So that was previously. And so now for this work, we took those standard leukemia stem cell markers, and within that compartment of that standard leukemia stem cell, cell surface protein, we sorted CD69 high, CD69 low, and actually we had some other cell surface markers, but just to simplify the point. Yes, so then we sorted and put them into mice and did colony forming assays. So we sort of demonstrated it at the protein level, but like in mice in vivo. Okay. All right, so basically all of this work is just to demonstrate really that my lab was very interested in finding kind of functional states of leukemia stem cells. And so we felt, well, we could do this in our mouse model, and we had some work in primary human samples. We thought, why don't we attack p53 mutant AML? So the first question we asked was a really like a general one. Because we know that p53 mutant AML tends to relapse or relapses much more aggressively than any other leukemia, we thought, well, does this mean that the p53 mutant leukemia stem cell has a unique molecular state that we could potentially target with drugs? And so we asked, well, what are those molecular features? A very broad question. And so before we talk about that a little bit, I just want to introduce p53 a little bit. I realized a few minutes ago before starting this talk that whenever I give this talk, I don't talk about p53, and I assume that everybody knows p53, but just so we're all on the same page, p53 is probably the most well-described gene and protein in the human genome, and it is a tumor suppressor. It is known as the guardian of the genome. The most famous and kind of canonical functions of p53 is to induce apoptosis and cell cycle arrest in response to stress like DNA damage. This is sort of canonical p53 functions. And then there are, people have shown a lot of other like important functions of p53, but we think of p53 as a tumor suppressor, and we think that if you lose p53 function, you cannot, arrest your cells don't die, and that is why p53 mutant cancers are so bad. That's a general thought. But the story isn't really quite that simple, and I just wanted to mention this concept briefly. People have noticed that mutations in p53 seem to give a pro-oncogenic gain of function property to p53. So, and that gain of function is not to make a super strong p53. It is a neomorphic gain of function. So it's gaining new functions, not, so a gain of function p53 is not like a really good tumor suppressor. It is not a tumor suppressor, and it is thought of by some people as an actual oncogene, and some people think it has a dominant negative function, and we won't get into that here, but simply just to point out that in AML and other malignancies, most of the mutations in p53 are missense mutations in the DNA binding domain, and there's really good data to show that these mutations allow p53. So these mutations prevent p53 from binding DNA, but they allow p53 to bind novel protein partners that lead to novel gene expression, and this novel gene expression leads to these pro-oncogenic effects that have been described in various models. Okay, so when we're talking about AML, p53 mutant AML, there is good reason to think that those mutant p53 genes are giving us new functions in that AML. And this is really, really important because it gives us a handle to think that maybe those new functions are something that we could inhibit therapeutically. Okay, so when we think about AML and other malignancies, we also think about signaling pathways, right? So we know that signal transduction pathways are widely hyperactivated in a bunch of malignancies, including AML. And almost every one of those drugs that I showed you in that first slide are targeting a signaling pathway. And these signaling pathway inhibitors are excellent drugs. And so we really like to think about signaling pathways. Okay, so when we set about to try to understand, are there some ways that we could potentially target p53 mutant leukemia stem cells, one of the first things we did was just scour the literature to look for drugs that might have some activity in p53 mutant AML, and we got all of the p53 mutant AMLs that we could get from our leukemia bank, and we treated them in vitro with this panel of drugs. So it was a sort of a shotgun effect, and what can we get? So we found a few drugs that seem to reduce leukemia stem cell frequency and also completely abolish colony formation in vitro, or you would call it maybe ex vivo, in these primary human AML samples. And so these are the drugs that we kind of called leukemia stem cell targeting inhibitors. Now, I don't want to get into it too much, but each of these drugs has a different kind of predicted target, so different signaling pathway target that it inhibits. And so we wanted to know, is there some kind of common pattern to the drugs that seem to work, are what we call the effective inhibitors. And so we took to a technique called CyTOF, or mass cytometry, which allows us to profile the intracellular signaling activation state of leukemia cells. And so CyTOF is basically like flow cytometry. So we could like measure all the cell surface proteins so that we can find the stem cells, but it also allows us to look at the single cell level at intracellular signaling pathway intermediates, and we can look specifically at the phosphorylated forms of these intermediates. And so we could just look at all the signaling or many signaling pathways within stem cells. And here I'm showing you one such plot. So this is a Visney plot, and the way Visney plots work is that they, based on cell surface protein expression, they kind of, all the cells that have similar cell surface protein profiles are kind of arrayed next to each other. And then the color of the plot is represented by, whatever you want to measure. And here we're looking at NF-kappa B levels. Okay, so red, it means high NF-kappa B levels, and yellow would be lower, and blue is low. So, and here's, and this is a representative primary human AML sample. When we treated with Venetoclax, and some of you will recognize Venetoclax as a BCL2 inhibitor. It is among blood cancers like our favorite new drug. It works really, really well in AML and CLL and a bunch of other drugs, and we just give it like, water to basically anyone who walks in the door. Anyway, so we use it, and guess what? It doesn't work very well in p53 mutant AML. And when we look at a p53 mutant AML, and we treat it with Venetoclax, we do not see a drop in NF-kappa B. But when we treat it with some of these, what I refer to as effective inhibitors, the inhibitors that drop leukemia stem cell percentage in our primary samples, we see a loss of NF-kappa B. And this is just it's a representative slide, but basically all of our effective inhibitors lowered NF-kappa B in our samples, and the drugs that did not work, the drugs that did not lower leukemia stem cell percentage in our P53 mutant samples also did not lower NF-kappa B. So we became a little bit, focused on NF-kappa B for this reason. But then we wanted to ask us a different question, which was, again, just a broad profiling question, which was just what is the signaling activation state of P53 mutant leukemia stem cells? And so here we went back to the CyTOF and we compiled a bunch of these primary human AML samples, some of them with P53 mutations and some from other genetic subtypes. And here I'm showing you, this is actually the levels of these signaling intermediates within the leukemia stem cell of each of these subtypes. So we're just comparing leukemia stem cells across subtypes. One thing you'll notice here, Ki67, you might recognize as a proliferation marker. The leukemia stem cells of a bunch of different AML subtypes have lower Ki67 levels than the sort of bulk leukemia sample. And this has been very well described in the literature that leukemia stem cells are characterized by being poorly proliferative relative to the bulk tumor cells. So that kind of was sort of reassuring to us. We expected to see that. But what we noticed right away, and you might notice that these red bars, these P53 mutant samples, have high levels of NF-kappa B. They also have high levels of 4-EBP1, you might notice. So we found within these samples this unique hyperactivation of NF-kappa B, which was interesting to us in combination with the previous work where we showed that the drugs that the drugs that target leukemia stem cells in P53 mutant AML seem to lower NF-kappa B levels as well. Okay, so we looked very specifically and we compared NF-kappa. So then we went back to our original work, right, where we showed that CD69 protein expression on the cell surface seems to identify a leukemia stem cell more precisely. And when we looked at P53 mutant AMLs versus other AMLs, we see that NF-kappa B is actually higher in those CD69 high cells in the P53 mutant relative to the other AML subtypes. Okay, so then we got more interested in CD69 as perhaps a unique marker of self-renewal, uniquely, let's say, effective marker of self-renewal in a P53 mutant AML. And so we asked the same question that we asked in the mouse, but now we're asking it in primary human samples. We took a bunch of primary human samples, we sorted now only on CD69 levels, and we see that the CD69 high sample, so each of these represents a different AML sample. So high CD69 levels identifies a subset of human AMLs with enhanced colony forming capacity. And it's really only the ability to form secondary and tertiary colonies that defines a stem cell. But you'll notice that some of these samples didn't even allow us to do serial colony forming assays because we couldn't even get primary colonies from the CD69 low compartment in some cases, but we did take the ones where we could get primary colonies, these guys. And so we took these guys and we plated them in secondary colony forming assays, and we couldn't get any secondary colony forming assays from the CD69 low compartment, demonstrating that that CD69 high compartment in primary human AML samples identifies the subset with self-renewal capacity. So to put this all together, we felt convinced that this data suggests that in a mutant P53 leukemia, hyperactivation of NF-kappa B is likely an important driver of self-renewal and could also offer a target for drug treatment. And so and I don't want to get into this too much, I think, because, well, it's highly detailed, but there's a bunch of literature and some of our own work that suggests that perhaps mutant P53 through that gain of function might hyperactivate the mTOR proteosome axis. And the reason that was super interesting for us is that proteasome inhibition is a very common way that we clinically and also in research inhibit NF-kappa B. There are no clinically effective NF-kappa B inhibitors, and so inhibiting the proteasome is sort of how it's done. And the proteasome is also very attractive to us because proteasome inhibitors are very safe, well-tolerated drugs that we use like all the time in some other heme malignancies. So as a heme malignancy, clinician, this is a drug I love and enjoy. So, and my patients love and enjoy it too. So we really got interested in this access as a possible treatment. Oh, sorry. You know what? I just want to tell you one other thing. I think anyway, I think I put this slide in late and I apologize. One other thing we really wanted to demonstrate and pin down is this potential relationship between P53 mutation, hyperactivating NF-kappa B. And so we took a P53 mutant AML cell line and we knocked down P53. And here we're looking at CyTOF data. Again, when you knock down P53 in this orange curve, you lose P53 protein. We also saw it at the gene expression level. And then we saw a loss of NF-kappa B. So that sort of cemented our kind of belief in this access, and then we asked another question. So I apologize. The proteasome story comes in a minute. I lost myself here, but anyway, so going back to this axis to ask, does mutant P53 really hyperactivate NF-kappa B and the proteasome? We showed that it can happen in this cell line, in this P53 mutant AML cell line, but we thought if this is true, if this is really happening in human patients, we should be able to just look at the gene expression data sets. There are like hundreds and hundreds of these patients published, and these data sets do include P53 mutant samples. And so here, this was done by a graduate student in our lab, Ryan. He compared the gene expression profiles of P53 mutant and P53 wild type AML. This is a Beat AML data set, but we did it actually in a totally unrelated data set, the TCGA. We saw the same exact results. And what I'm demonstrating here is gene set enrichment analysis, where we see the NF-kappa B gene targets or gene sets, and they're all the 19 gene sets that we found in that data, in the molecular signatures database. They are all enriched in P53 mutant AML, and they all have very significant FDRQ values. So these are like your p-value that's stringently corrected for multiple hypothesis testing. This is really very convincing data. And there were fewer proteasome gene sets, but also same thing. So proteasome gene targets and NF-kappa B gene targets are all enriched in P53 mutant AML samples from patient databases. And so this is just a representation of some of that, the GSEA data showing NF-kappa B and proteasome enrichment, although this one has a lower FDRQ, but most of these were pretty significant. We also went to another data set. This is the CMAP data set. So that data set is kind of complicated, but it basically shows the gene expression profile of certain inhibitors in cell lines, okay? And so the P53 mutant AML samples gene expression profiles look like they are being treated by proteasome inhibitors. They look like I'm explaining this badly, but basically looking at what it looks like to be treated by a proteasome inhibitor or NF-kappa B inhibitor suggests that P53 mutant AML samples have higher proteasome and NF-kappa B genes, basically in that data set based on inhibitors. Basically, all of this put together, when you look at the gene expression data, suggests that NF-kappa B and the proteasome are upregulated in P53 mutant AML. And so here, we get to the final point, which is we tested the effect of proteasome inhibitors on P53 mutant AML. And we saw that when you treat primary human AML samples with proteosomes, so these are proteosome inhibitors, these are 3 different proteosome inhibitors, you see loss of NF-kappa B in the leukemia stem cells, you see loss of primary colony formation and secondary colony formation from each of these inhibitors. Basically closing out this part of the talk, showing that we think that mutant P53 is upregulating the proteasome, possibly through mTOR, possibly directly through proteasome gene upregulation, and that upregulates NF-kappa B, and that this hyperactivation of NF-kappa B is central to self-renewal in a P53 mutant AML, and that this might be a really fruitful area for effective targeting in P53 mutant AML. And almost all the work, except for what I showed by Ryan, was done by Marie Lou Antony and Clara Noble Orkut in our lab. Hi, Erin. Come on in. All right, so then I want to show you another story of the way we're attacking this problem. And this story takes us back to another malignancy called myelodysplastic syndrome. We call it MDS. It is considered a pre-leukemic state, even though in itself it is a malignancy. It causes bone marrow failure, but the big clinical problem with myelodysplastic syndrome is progression to AML. So if a patient has MDS, what we're deathly afraid of is that they're going to progress to AML. And if they do progress to AML, their outcomes are horrible, with survival rates again in the months range. So of course, we think about P53 mutant AML. MDS is the worst of all the MDSs, and they are much more likely to progress to AML. And so once again, P53 rears its ugly head. The thing that people have noticed, though, is that, sorry, here's the survival. The likelihood of transformation to AML in a P53 mutant setting is really much higher than in a wild type setting for MDS. But what you'll notice is that this doesn't really achieve 100%. So not all P53 mutant MDSs will progress to AML. And so we thought this could be a really good model to understand the progression, how P53 mutations cooperate with perhaps other genes or other events to progress MDS to AML. And so actually, this is before I got involved in this whole topic. David Largaspada made a mouse model where, here we go, where he took common MDS genetic events like P53 mutation and coupled it with sleeping beauty mutagenesis. And just briefly, sleeping beauty mutagenesis is a kind of random mutagenesis where the sleeping beauty transposon will insert in the genome, and depending on the orientation of insertion, will either cause gene upregulation or gene disruption. And if sleeping beauty inserts into a gene that gives the cell an advantage, that gene will cause outgrowth of leukemia. So that was the concept, that if we get interruption of genes that can cooperate with P53 or with the mutant P53 to induce progression to AML, we will get AML. So this was a way for us to look at basically cooperating genes and look at mechanisms of progression to AML. And the cool thing about this model, obviously, is that it is immune competent, unlike our AML patient-derived xenografts. So we took this mouse and we just looked at what happened in this mouse. And basically we found that in mice that did not have sleeping beauty mice that did not have sleeping beauty. So if you just have a P53 mutation with a deletion of 5q, another common MDS abnormality, we didn't see any progression to AML. If you just have sleeping beauty, we saw very few AMLs. But when you took this P53 mutant mouse or the deletion 5q mouse and layered on sleeping beauty mutagenesis, you got a lot of AML. So we thought these mice could teach us a lot about progression, mechanisms of progression to AML. And I just have a tiny one, I think one slide or 2 slides on this topic. So we did gene expression profiling and looked for fusion genes. So where this sleeping beauty was inserted, which genes sleeping beauty was inserted, and you'll see here all these red boxes are insertions, and each of the columns is a different leukemia. And you see this just sort of row of red being ERG. So this was really interesting to us. We never saw ERG in any of the Sleeping Beauty tumors, and we saw it in almost every one of these other tumors, AMLs. So ERG it was interesting to us. It hasn't actually been well characterized, and it is not on anyone's radar for myeloid malignancy, but there was a paper that just came out this summer that showed that if you look at secondary AML, so second AMLs that progressed from MDS, you see a lot of them have ERG amplifications. So it's not a mutation, so it never made it into the database, but it is amplified in human samples, and nobody really knows what it does. So here we have these beautiful models that we can use to understand what ERG is doing, how it's contributing. And you notice also, we notice that ERG is often hyperactivate or disrupt or probably upregulated with this other gene, and this is ERAS. So that is also something that we're interested in pursuing. And then another, not as common, but also commonly disrupted gene is the IL-2 receptor beta. And this is interesting because it is implicated in immune evasion. And we know that P53 mutant AML is particularly immune evasive or suppressive, I guess I should say. So this is where we are with this project. This project is being led by a graduate student in my lab, Daniel Chang, and Alpay and Aaron have been really very strongly contributing to this project. And of course, we're doing this in collaboration with David Largaspada. So here we have this immune competent model where we can really study the stem cells and how these fusion genes are interacting with P53 in stem cells of a P53 mutant leukemia. Yeah?
You probably said this when you produced the model, but is that maybe?
I did not say that, but thanks for asking. So it is actually a full body P53 mutation. These leukemias were just, and the SB is under an MX1 CRE promoter. Those bone marrows were transplanted into recipient mice, and then they were fed PIP-C. And so in this setting, it's a syngeneic model, but only the bone marrow carries these mutations. But it is kind of the full body P53 mutation allows us to ask some other questions that we're sort of really interested in also, that we're working on kind of, I would say, in parallel. Actually, we know, so most of the mouse models that have been published in P53 are either, organ specific and almost always knockouts. So there are like 2 mouse models that have been published in AML that have mutations in P53. Everything else is a knockout. So all the work is always knockout. And so we think that a lot of the work is missing that like gain of function, function of P53. And so we're actually just also characterizing the bone marrow of that mouse. Like does that mouse get MDS when it's old, for example, that kind of thing. So anyway, yeah. Okay, so that's where we are with this. It's kind of a new project that we're actually super excited about, but we're early days. And here I'm going to show you one last thing that we do, and this is a totally kind of different question that we're asking. And here, this is a purely bioinformatics project. So I really enjoy this kind of thing. So, and this is really thinking about the fact that we have, all these hundreds of AML patients where we have gene expression profiling and correlated clinical results. And can we learn anything just by looking at the data that's already there? And I should say, when I first started my scientific training as a high school student, working in somebody's lab for the summer, he told me, you can't find something new by using the same old microscope everyone else has. So I always had this thought that we need new methods to like sort of move science forward. But I always thought also that, we have like just all this tons of data publicly available, and can we get more from it? And then the other thought I had was, well, P53 mutant AML is really, really, really bad. Like, it is uniquely very bad. The outcomes of those patients is so much worse than any other AML patient. Is there something that we can learn from the gene expression profiling? And in ALL, which is a totally different disease, acute lymphoblastic leukemia, somebody had that idea a very long time ago. And in ALL, the bad actor is the Philadelphia positive ALL. And many years ago, a couple of groups identified that if you have the gene expression profile of a Philadelphia positive ALL, those ALLs behave just as badly as a Philadelphia Philadelphia negative. So if ALLs without a Philadelphia chromosome, but that have the gene expression profile of a Philadelphia positive ALL, those Philadelphia negative, but Philadelphia-like, they're called ALLs, behave just as bad. And that's what we're looking at here. So in this last bar here, this is the survival of patients with a BCR-ABL, the Philadelphia chromosome. Those ALL patients don't do very well. If you have the gene expression profile of these leukemias, even though you don't have a BCR-ABL, you do just as badly. And if you don't have either of those gene expression profiles, you do pretty well for ALL. And so these Philadelphia-like ALLs were recognized as a clinical entity, and there are clinical trials to try to treat these patients better. And so recognizing that at the gene expression level, we can identify a Philadelphia-like leukemia and that we can do something about it was sort of established in ALL as an important paradigm. And I thought, well, why don't we see if that could be the case for AML? So that was my naive question that we asked this question a year ago. And so to do this, this is a pretty complicated bioinformatics undertaking that Ryan in my lab did, and really it was Chad Myers that designed everything about how to ask these questions. So this is really an exciting collaboration for me. Basically, Ryan took one of these data sets, divided them in half into a test and training data set, and used ridge regression analysis to train this model to define a P53 mutant gene expression profile, and then tested it on the other half of the data set and validated it on this other unrelated data set. And sure enough, Ryan with Chad Myers identified a group of human AML samples with a gene expression profile of P53 mutant AML, but that these samples, even though they have the same gene expression profile, they don't have P53 mutations. They are P53, we call them mutant-like. Okay, so then we asked the important question, can these leukemias with that P53 mutant-like gene expression profile, do they have a different survival? And they do in these two independent gene expression profiles, independent data sets. P53 mutant-like AMLs have worse survival in these two different data sets. And some of you, the 2 or 3 of you in the room who do treat AML patients will recognize, I think this was, to me, very important. We segregated the data by ELN classification. So the ELN, the European Leukemia Net, allows us to predict outcomes in AML based on the genetics. And so we have favorable, intermediate, and poor risk subgroups in AML. And we treat those leukemias differently using different strategies. And basically, and the numbers are pretty small by the time you do all this subsetting, but for these favorable risk, also for the other risks, but to me it was really significant. For these favorable risk leukemias, if they have that P53 mutant-like gene expression profile, their hazard ratio for death was much, much higher in 2 independent data sets, suggesting that this gene expression profile sort of supersedes any other classification system that we have. So these are patients, the favorable risk patients are patients who would not be offered transplant. We expect to cure them in first remission with just chemotherapy. And in fact, if they have that P53 mutant gene expression profile, we won't. Their survival will probably be worse. So this, to me, was a particularly kind of chilling finding. We also just asked, what are these mutant-like AMLs, sort of using our common clinical parameters. And the reason we wanted to ask this question was a little bit thinking to the future. if we want to get these patients on upfront clinical trials. With AML, that's always really hard because usually a patient comes in with AML, you need to give them chemotherapy within a few days. You don't have time to wait, 2 weeks for a clinical test. And so we thought, do these have clinical features which would suggest that perhaps they can wait for us to do this kind of clinical testing to see if they qualify? And basically, we looked at bone marrow blast percentage and white blood cell count of these patients when they were first diagnosed, because the higher your white blood cell count and blast percentage, sort of the less time you can wait, and the lower your white blood cell count, kind of the more you could wait to start therapy, basically. And what we saw, and this has been sort of published in the literature, but not really focused on, it's not something that's well recognized, but in 2 separate independent data sets, P53 mutant AMLs tend to have lower bone marrow blast percentage and lower white blood cell counts. These are, and clinically, I think I already sort of knew that. These patients come in and their leukemias are not these like rip-roaring, raging leukemias that are about to kill them in the next few days. This is, the P53 mutant AML is more of a slow burn kind of AML. It's not the one that comes in with a super high white cell count or bone marrow blast percentage. So we saw that. This is my experience clinically, but also we saw that in the data sets. And sure enough, the P53 mutant, like AML, shares that feature, that the white blood cell count is a little bit lower, the blasts are a little bit lower. And in fact, this spurred like a chart review we're reviewing our own experience with AML and looking to see what our AML patients look like clinically. But just to kind of finish this story with a little more detail, that Beat AML data set also did ex vivo drug screens for all like 400 leukemias and about 120 drugs. And this blue, so this is a heat map showing drug sensitivity, and blue represents resistance in ex vivo to these drugs. And you see that P53 mutant AMLs are largely resistant to every drug they tried, and the P53 wild type had a lot of sensitivity, at least at this drug screen. And the P53 mutant like AMLs look like P53 mutant AMLs in that they are largely treatment resistant. There is this kind of important difference that might be important to explore in the future, but I do want to highlight one thing here. So this little bar is Venetoclax. Remember, that's our BCL2 inhibitor that we give to almost every patient. You can't die of AML without first getting some Venetoclax. And sure enough, the P53 wild type patients are pretty sensitive to this drug, at least on this drug screen. P53 mutants are resistant, and P53 mutant like AMLs are very resistant to this drug. And one last thing we did then, so I said to Ryan, okay, so this is all very exciting, but like I told you, this is only important, this is only relevant if I can identify these patients and put them on a clinical trial, otherwise it's useless. So give me a gene list, give me a gene signature. And so this was quite some work because 2 different data sets, we needed it to work in 2 different data sets, and we needed it to be really robust. And I was thinking here of like an Oncotype DX kind of test, like we do for breast cancer, like a short gene expression signature that can identify these patients. And sure enough, there's a 23 gene list that has a really nice sensitivity across 2 separate databases. So we have a 23 gene signature that can identify these P53 mutant like AMLs. And what I would hope for and envision in the future is that we could have clinical trials that would enroll P53 mutant AML patients and P53 mutant like AML patients in the future. Okay, so sorry, I kind of rushed at the end, but we got through, we talked about some signaling or pathways that we could use to target leukemia stem cells in P53 mutant AML, a model to study progression, mechanisms of progression, and then some gene expression data that we did that obviously sorry, I didn't mention it, but this project with the gene expression analysis is a rich source for other pathways and cell surface markers and all this other kind of thing to understand these leukemias. Really, all of the work that I showed just about was done by Clara Noble Orkut. I'm sorry, I forgot to add, change this, I don't know where that came from, but Marie Lou Antony, Yoon Kyu, who goes by Ryan Lee, and Daniel Ching, with really important contribution by our collaborators, and of course the Leukemia Bank, the Heme Malignancy Tissue Bank, which is managed, largely by Veronica Bacanova, is absolutely instrumental to our work. So, thank you.
Let me find a microphone here.
All right, well, thank you very much. I have a virtual question. The question comes from Jeremy Allred. Do your data sets discriminate between monoallelic TP53 alterations versus biallelic? Do you think that this matters on your NF-kappa B axis, or is it a gain of function interaction with the mutation?
That's a really, really good question. No, we don't. Most of the NGS, like clinical grade NGS, doesn't really allow you to do that. There are cases where it would, but that's not easy to do. We do know that about 70% of P53 alterations in myeloid malignancy, so like AML and MDS, will be biallelic, meaning that for one reason or another, you do not retain the wild type allele, but we don't know specifically for these leukemias. I know that Daniel did look into our mouse leukemias, and I believe he was able to identify wild type RNA, at least in that mouse model. That is something I've been talking a lot about to the molecular lab, at least for so some of those leukemias that I showed in that first part, we have engrafted in mice, and we have a panel of these patient-derived xenografts, and we did look for that, the biallelic targeting in some of those. And in fact, we identified 2 with biallelic knockout of P53, so that's pretty rare in AML. It's, I think, maybe 10% of AMLs actually have a knockout of P53. We have 2 PDXs like that, so those are really great controls for everything we're doing.
Here you go.
I was curious. Oh, thanks. I was curious about your list of genes, and does any pathway jump out? Or are they cell cycle? What are they?
Yeah, so that's a really good question. So we haven't done pathway analysis on these genes. We've done pathway analysis on like the greater
Largers.
and like what you thought would jump out did jump out. Actually, not so much cell cycle, but more like some cell cycle, some DNA damage, all that kind of thing. But NF-kappa B, proteosome, a lot of like mitochondrial, what's the word, metabolism kind of gene lists are at the top. I do want to point out this gene right here, this CREB-BP. So that gene, for example, was recently identified as being involved in a translocation that has just now made it into ELN classification. So if that gene is involved in a translocation, it's now newly identified as a poor prognostic indicator for AML. So anyway, but yeah, we do have all that pathway analysis.
Yeah, well, I was just thinking, if you're going to be P53 mutant-like, but wild type P53, it's going to be, to me, it's going to be proximal players that are maybe binding partners with P53, are very close associates of P53. That's why I was kind of scanning down that list. I don't see any, but that's cool.
Yeah, but I think we also have to keep in mind that the missense mutations, the DNA binding domain mutations, seem to have novel binding partners. So not what you know what I mean? So that's been nicely worked out, mostly a lot in breast cancer, actually. A lot of really good literature in breast cancer, a little bit of literature in murine AML. We're hoping to figure this out in primary AML.
For the drug screen that you did to identify drugs that were effective against the leukemic stem cells, did you also test the activity of those drugs in your non-leukemic stem cell population? So were they uniquely effective in the stem cell against the stem cell compartment, or just broadly effective across the board?
That's a really good question. We did not look. We haven't looked at that. And I know that's a really kind of common question to ask. Are you targeting, are you like just sort of ablating the bone marrow or not? I think those would be really good experiments to do. In my mind, they were, you know, if we had all the money and hands in the world, we would do them. But in my mind, in AML, we do ablate the bone marrow and we're just fine with that. You know what I mean? So it felt like, yes, we would want to know that, but first we just need to get rid of the leukemia. And if we need to ablate the bone marrow for it, that's okay. We're prepared to do that.
And did you look at those drugs and other assays to identify their mechanism of action? Is it actual cell kill, or
Oh, I see what you're saying.
is it doing other things?
Yeah, that's a good question. We haven't done that. I think that would be, so, you know, originally we just sort of did that screen and then we kind of focused on like, can we just go to the proteosome and focus on that? So we sort of left that panel of drugs behind and tried to find something more kind of streamlined. We have done a few apoptosis and like ROS and those kinds of assays, but we haven't done a lot of that. I think that would be like an important thing to do also once we've sort of laid down our, you know, kind of identified our favorite drug and.
Or even with the proteasome inhibitors, if that's where the focus
Absolutely,
is,
yeah.
instead of working with the upstream drugs.
Yeah, no, absolutely.
Zohar, great talk. I was really interested in the fact that the SB screens you did were done with a transplanted population to get started into a population. Were they done from the same founder marrow population, or are there differences in that population which had P53 activated already before you turned on Sleeping Beauty that may push them down one leukemic pathway or another?
Yeah, there were a few founders like per genotype, like a few, not like on the order of 3 to 5. This, I think, are you getting at the question of clonality? And I think that's a terrific question. And in fact, we are retransplanting these leukemias into recipients to try to expand them and ask a little bit more of a question about clonality and clonal evolution. But yeah, we don't have any of that data yet, but that's very important.
Zohar, great talk. So I have maybe 2 questions.
Yeah.
On the first part with your proteasome inhibitor in P53, you're saying that inhibiting the proteasome kind of downregulate NF-kappa B, right?
Yeah.
Have you looked at the P50, because P53 is only also kind of like being degraded, and kind of in the model for P53 is that the function of P53 is need to be not degraded in order for to kind of promote cellular function. So how have you thought about that in terms of what is the P53 protein function would be when you treat with proteasome inhibitor?
Yeah, I mean, that's a good question. Right. So you're right. P53 is wild type P53 is controlled by directing it for degradation, and wild type P53 levels are kept low under normal steady state conditions, right? And then you have some kind of DNA stress or whatever, and that's relieved, and P53 protein is upregulated. In a P53 mutant setting, almost exclusively it is resistant to proteasome degradation, and that's why actually being able to detect P53 protein is a proxy. It's a really good like clinical proxy for P53 mutations. But that is a good question. If you inhibit the proteasome, are you further upregulating P53? We haven't thought about that. We should look into that. Like, are we further? So if mutant P53 is acting as an oncogene and inhibiting the proteasome further upregulates P53, you know, is that having an effect? That would be an interesting question.
Just to kind of figure out what is the clinical outcome.
Yeah. That's a good point. We do see, so that work with I didn't want to like get into like a big, but there's some really nice work that shows that a variety of P53 DNA binding missense mutations, these gain of function mutations, a variety of them bind NRF2, and NRF2 induces the upregulation of proteasome genes in a number of solid tumors. That was shown in a number of solid tumors. Of course, they did not look at leukemia, but then we looked at leukemia at those mouse data sets in AML, P53 mutant AML gene expression profiling, and indeed we see upregulation of proteasome genes as well. And so that's kind of the proposed mechanism for how that might work.
And then just a quick question on this last beta ML data sets, right? With these gene sets.
Yeah.
Thing is that when you look at the P53 like kind of signature,
Yeah.
are you able to potentially take the Beat AML's data set and look at AML patients that are de novo kind of AML versus MDS AML to kind of link it back to where your P53 status is? And then the other one is also, have you maybe correlate this with ENCODE data for looking at like transcription factors and seeing whether there's a certain signature of what transcription factors are binding?
Yeah, I mean, I think those are terrific questions that are still kind of on the list. This data is really, as I said, the whole concept originated a year ago. And so this is as far as we've gotten so far, but I think those are really very terrific questions. I think the next step is really to ask about, look at MDS, look at secondary AML. I think the secondary AML is going to be a little hard because it's a small subset of both the beat AML and TCGA, but right, we have this like really nice MDS data set that we could look at. Yeah, so I think that is a really important next question for these. I didn't mention this, but P53 mutant AML is fairly rare. It's about 15% of AMLs, but when we look at our AML patients, 30% of them have P53 mutations, the AML patients that come to our institution. So I think we sit in a really unique place to sort of work on some of this.
All right, great. If there's no more questions, we'll say thank you one more time to Dr. Sachs.
Thank you very much.